the last lecture, we went over glycolysis. We looked at how ATP is generated from glucose, what enzymes are involved in these 10 important steps, which were the important intermediates, which enzymes were regulatory steps and why. We looked at the delta Gs. And we looked at how other sugars fit into this glycolysis cycle, where they feed in and how they're broken down. So we've broken down glucose to pyruvate. And as you recall, we've only generated 2 ATP. So what happens at this point? Are there other fates for glucose and other 6-carbon sugars? What happens to them, and what happens to pyruvate? And that's what we're going to look at today. Okay, so one of the other paths for glucose is called the pentose phosphate pathway. And we're not going to go into this too much, but I just want you to be aware of where else glucose can go. This is driven by the need for the ribose 5-phosphate, which you remember is involved in nucleotides. So sometimes glucose is broken down into glucose 6-phosphate, but rather than go through the rest of glycolysis, it actually goes off on this other pathway, which is called the pentose phosphate pathway, and can be converted into ribulose 5-phosphate and xylulose 5-phosphate, and then can go down over here into nucleotide biosynthesis. Another place it can go, if the organism doesn't need that, is up into fatty acid synthesis. So if it doesn't need glucose for energy, which would be the main path, and it doesn't need glucose to make these 5-carbon sugars for nucleotide biosynthesis, then it shuttles whatever is necessary, whatever's left over off into the fatty acid synthesis. So the incidence of most of the common form of glucose 6-phosphate dehydrogenase deficiency is a 10-fold reduction in this enzymatic, and this is the enzyme in here. We're not going to go into the enzymes too much for this one. So it's a glucose 6-phosphate dehydrogenase, which takes the glucose over to the ribulose 5-phosphate. And this is characterized, or the deficiency is characterized by a 10-fold reduction in enzymatic activity in red blood cells. And 11% of African American heritage have this deficiency. This high frequency suggests that this deficiency may be advantageous under other environmental conditions. So people that had this deficiency were actually in better shape in the case of for African Americans, because this deficiency actually protects against malaria. So the parasites causing this disease require the NADPH for growth, and then we're not generating enough NADPH if we can't go through this part of the cycle and have this deficiency. So they were actually better able to fight off the parasite. So the flow of glucose 6-phosphate depends upon the need for NADH, as we said before, the need for the ribose, and the need for ATP. So there are a couple of different models depending upon to decide which way things go. Much more ribose phosphate than NADPH is required, or the need for NADPH and the ribose phosphate are balanced, or you need more NADPH and less amount of ribose 5-phosphate, or if they're both required. So all of these different scenarios determine which way the glucose can go. So besides from that, we said that glucose, we've gotten all the 6-carbon sugars down through glycolysis. And the ones we want to keep for something else went off to the pentose phosphate pathway. And now what? So we've gotten to pyruvate so far. But if you look at the amount of energy we've gotten, only a small fraction of the total available energy has been realized at this point. When glucose is oxidized completely in a lab to carbon dioxide and water, the standard free energy change is 2,840 kilojoules per mole. So that's quite a bit of energy out of 1 mole of glucose. The glycolytic degradation of glucose to 2 molecules of pyruvate has a much lower delta G, and it only yields about 5% of the total energy that could really come out of a glucose molecule. So the 2 molecules of pyruvate formed by glycolysis still contain a lot of energy that we want to get out. And this energy can be extracted by going further through oxidative reactions, which in the citric acid cycle, the carboxylic acid cycle, Krebs cycle. There's a bunch of different names for it. So we're going to see how we can take pyruvate to get more of this energy out. So Let's talk about the fate of pyruvate. There are actually 3 fates of pyruvate, depending upon the conditions. So the usual one is the aerobic conditions, and that's the one we're going to spend the most time talking about, but I'm going to get back to that. Under anaerobic conditions, you can create lactate, and there's also an alcoholic fermentation condition that is used by yeast. So the one that is really going to give us the most energy is taking pyruvate, converting it to acetyl-CoA, and then taking it through the rest of the TCA cycle. And we're going to go into that in detail. The alternate pathway involved in alcohol fermentation occurs in yeast, takes this pyruvate and takes it through acetaldehyde, and then onto onto 2 ethanols. So we can make we can reduce the pyruvate, because it has less oxygen over here. So this one is more oxidized. This one is less oxidized. We're going to reduce the pyruvate to ethanol and end up with some energy. The other pathway is under anaerobic conditions. The 3-carbon pyruvate is converted to lactate. And this is what happens during strenuous exercise. Oxygen is depleted and anaerobic conditions result in the muscles. Pyruvate is converted to lactate, and this increased amount of lactate is what causes your muscles to become tired and sore. After the exercise, you take deep breaths, you breathe in heavy, you're getting the oxygen back, and you can reform pyruvate in the liver, and then pyruvate will go through the regular part of the citric acid cycle. So those are the 3 fates of pyruvate. So once we're most concerned with the pyruvate that's going to go through the citric acid cycle, and this is going to go through a series of enzymatic reactions, including the enzyme, the pyruvate dehydrogenase complex, and that's going to take the pyruvate into acetyl-CoA. And you can see here this is a high energy reaction. It's irreversible, and it can release a lot of energy. This is what acetyl-CoA looks like. It's the acetyl group on the end here. It has this panthenoic acid vitamin in the middle, and it has this phosphorylated ADP on the end. And this whole thing together is called acetyl-CoA. So we have pyruvic acid, and joins up with the coenzyme A to make this acetyl-CoA, and then it will feed into the citric acid cycle and generate more ATP and more energy. So before I jump into the citric TCA cycle, we're going to talk about some of the other things that feed into it as well. So fatty acids, amino acids, and pyruvate all have this common intermediate of acetyl-CoA. And then acetyl-CoA has choices where it can go as well. It can either go into the TCA cycle, it can become ketone bodies, or it can become sterols and fatty acids and feed back into fat storage. So since we're talking about fatty acid metabolism feeding into acetyl-CoA, I'm going to do that first. I'm going to talk a little bit about that, and then we'll go into the TCA cycle. So fatty acid oxidation takes place in the mitochondria. It is responsible for providing energy into cells when glucose levels are low. Most humans store enough triacylglycerols in their adipose tissue to supply the body for about 3 months during starvation. So if you're ever caught in the desert or the woods someplace, you can go without food for pretty long. You can't go without water, but you can go without food because you have enough fat stored in your body to go for about 3 months. Liver and adipose cells convert excess acetyl-CoA into fatty acids that can be stored. So you either have lipids from wherever they're coming from, or you're eating fats and lipids as well, and you need to break those down. So the next stage, we're going to look at breaking down of lipids and how they will eventually feed into the citric acid cycle. So fats and lipids provide an efficient fuel storage. The advantage of fats over polysaccharides is that fatty acids carry much more energy per carbon because they are more reduced. So this is the reduced side. You see, the more hydrogens you have on something, the more reduced it is. The more oxygens you have, is the more oxidized it is. So fatty acids are more reduced, so they have more energy per carbon than their counterparts as you get more oxidized. They carry less water with them because they are nonpolar. So where glucose and glycogen are good for short-term energy needs and quick delivery, fatty acids are for long-term, months storage of energy, and they have more of a slow delivery. This is looking at more details, more numbers for the oxidized and reduced. So the most reduced ends we said was when they're completely hydrogenated. So these have 820 kilojoules per mole, and as you take the hydrogens off and replace them with oxygens and they become more oxidized, then there is less energy. So in fact, carbon dioxide is all the way to the end. You're not going to get any more energy out of that. So you can take this saturated fatty acid and keep reducing it, and you're going to get more energy than you would out of just a glucose molecule, because that has more of these OH groups. So these long-chain fatty acids are the ideal way to store fuel for the body. It holds more calories, and the body has an unlimited capacity to store fat, as we all know, right? So you can always, very easy to gain weight, a little harder to lose it. So a 70-kilogram man with about 11% body fat, which is probably pretty low, has over 100,000 calories, kcals, stored as fat, but only about 600 kcals stored as glycogen. And the body can support life for a long time, 60 to 90 days without food. Okay. So 1/3 of our energy needs actually comes from these triacylglycerols. 80% of the energy needs of a mammalian heart are met by the oxidation of these fatty acids. Hibernating animals such as grizzly bears or other animals that hibernate through the winter rely almost exclusively on fats for their energy. And that's what happens over the winter. They're starting to burn off the fat that they have had stored. And camels, some animals, store fat as an eventual source of water. We're going to look at this at the end, but camels do not store water in their humps. They actually store fat that can be converted into water. So let's just talk a little bit about the digestion of fats. I just want to see how long I have. I think I'm at about 11 minutes. I may have to break this soon. So first we're going to look at the digestion of fats. There are 4 different parts to it. We have the fatty acid transport, beta oxidation, and then again feeding into the Krebs cycle. So we're not going to look at the Krebs cycle all at once at the end. So the first thing to do is to break down the triacylglycerols into fatty acids. And if we need any of those fatty acids, those are going to go off into making membranes. They're going to go off into making cholesterol if that's needed, and maybe ketone bodies if that's needed. And then eventually it will go into the TCA cycle for energy. So a small amount of digestion occurs in the stomach due to the lipases that are in the stomach, but most of the digestion is really in the small intestine through the secretion of these bile salts, and we'll talk about those in a minute. And then the pancreas secretes some more enzymes that secretes again into the small intestine to break down fats, and then most of this is happening in the small intestine. So emulsification begins in the stomach, but does not really take place until the bile salts break these fat particles into smaller cells in the micelles, into smaller micelles in the small intestine. So emulsification starts in the stomach and continues in the lumen of the small intestine. So what starts out as a big blob of fat has a limited surface area to attack, to break down. So in the stomach, the fat is heated to liquefy, peristaltic movements kind of break it up into smaller pieces, and then in the small intestine, these fat particles are coated with bile salts. So there are 3 enzymes involved. Pancreatic lipase, or triacylglycerides, breaks them into fatty acids. Phospholipase A, which requires bile, and it breaks down the phospholipids. And lipid esterases, which hydrolyze the cholesterol esters. So these are the 3 enzymes that we're going to have to know. So bile salts are amphipathic. That means they have a polar end and a nonpolar end. They are synthesized in the liver. They are stored and secreted by the gallbladder, stored in the and made in the gallbladder, and then secreted into the intestines. They act as detergents. They can break up the water-insoluble lipids. And the best picture is, oh, where's my picture? Is this picture where fats and waters tend to separate. Enzymes are always aqueous, so they're in the water portion of the cell. But the fats aren't, right there, because they don't dissolve in the liquid as well. So bile salts are amphipathic, so they have a part that will bind to the fatty part and also a part that will dissolve in the liquid. So they can bring the enzyme and the fat part together. So these small droplets of emulsified fat now have access to these enzymes. So there are various different lipids are used as transported as lipoproteins. VLDPs, which are the very low density, LDLs, which are low density, HDL, which are the high